You're listening to Weird and Dead, the paleontology podcast that tells evolution's most embarrassing and bizarre stories. I'm Megan Wetherell.
And I'm Amy Atwater. We are two living scientists who are weirdly excited about dead things.
On this episode, we discuss all things mummified and freeze-dried, except for people. We don't care about them.
It's true. But we will discuss all kinds of insane preservation, including pregnant horse placentas, dinosaur skin, ancient dinosaur protein, and we'll debate the fine dining experience of eating Ice Age bison.
Like always, our podcast talks about some disgusting things, so definitely don't eat while listening to this episode, even if they did. And check the warnings in the description. You can't talk about amazing preservation of soft tissue without talking about Messel, Germany. So 40-some million years ago, a volcano there blew up and formed a deep crater that then filled in with water. Fortunately for us, but unfortunately for the animals that were around at the time, the volcano didn't disappear. Instead, it burped out plumes of carbon dioxide at regular intervals, which killed animals and preserved them to an absolutely exceptional degree. One of the amazing things that this gassy pool preserved was horses, not like the ponies of today, but little guys that were tiny and adorable and had a perfectly normal number of toes. There are about 30 horses that are preserved there, and about 15 of them were female, which we can tell because of their childbearing hips. We can also tell that about 8 of them were pregnant because of, right? Like, oh, so cute. Dead babies. But it's not just horsey bones and their sad dead babies. In the region of the fetuses are these really strange black halos, which we now know are straight up preserved placentas. Well, kind of, we'll get to that, but that's how insane the level of preservation is at Messel. We're talking about whole organs getting preserved, which is not normal, by the way. Usually only hard things get preserved as fossils because if you take something soft and you squish it, it squishes, or it gets eaten away by animals and bacteria. But today we're going to talk about all the odd cases where that doesn't happen and what it takes to get those gushy bits to stick around. And Messel is an incredible place for that because it's not just pony placentas. It's hair on primates. We have amazingly preserved feathers that we can reconstruct color from.
Whoa.
We have amazing skin impressions. We have bat wings. So all of these like insanely detailed pieces of preservation. And while that's all at Messel, that is far from the only place that you get soft tissue preserved, as weird as it seems.
Those are wild. But that's fascinating. And bat skin, too. Wow. Bats are fascinating, too, because it's like they have a weird fossil record because it's like we don't really know what group they evolved from because it was like bats weren't there and then they were there with full-blown wings. And you're like, whoa, how do you just... And aren't some of the earliest bats from this site in Messel?
I think so, yeah. Some of our best preserved bats are certainly from Messel.
And we've got some good ones preserved in the Green River Formation of the
Yes.
Western United States as well. So
weirdly, both sites have pangolins. So I'm just going to go ahead and say that bats and pangolins have been hanging out for all of prehistory and are clearly a proxy for the other animal. They've been around planning COVID for millions of years, I think.
I was going to say that proves their innocence, that they just got co-opted into this as you're here. And it's like, we've always been here and we're always together. Don't point this at us.
Somebody needs to make that into a comic strip, is just bats and pangolins planning the world's demise.
Oh my God, it'd be so cute. Like 2 of the most misunderstood mammals out there. Mm-hmm. Someone.
Yeah, so tell us about some other stuff that has soft tissue / organic tissue preservation.
Yeah, so you're talking about a site that's about 50 million years old. What's really cool is that we have soft tissue preserved even older than that, too. We've got dinosaur soft tissue preserved as well as younger stuff, too, like more Ice Age, Pleistocene material. Those are pretty different, though. Like the Pleistocene stuff is usually frozen, and we're going to talk a lot more about that at the end. However, dinosaurs, which is wild to think about preservation of soft tissues in something that's at least 66 million years old. And in fact, it's out there. There are lots of examples of dinosaur skin that has been preserved, from Triceratops to Thescelosaurus to all sorts of hadrosaurs. I mean, there are lots of examples of the skin impression. And again, like while we say, it's essentially like the impression, right? It's not like the skin itself. It's just that the preservation was good enough that obviously the skin didn't decompose fully before mineralization came in. Which is pretty fascinating. And in fact, there are a couple examples of dinosaur mummies known out there too, which is really strange. And when I first learned about them, I was like, oh my gosh, there's like straight up mummified tissue. And no, it's more like dinosaur mummies that were fossilized is really what I've come to realize, because I never cared enough to Google it, but I did lately. One of the most famous and like best quality fossil dinosaur mummies is Leonardo, which, as I was telling Megan earlier this morning when I was thinking about it, I kept calling it Alejandro. Leonardo was found in, I think, around the year 2000. It's from Montana. This is another Brachylophosaurus specimen. So, man, they preserve well. I got to give them mad props. So, Leonardo, see, almost called him Alejandro again, is like, oh my gosh, something like it has the Guinness Book of World Records for the best preserved dinosaur. Over 90% of Leonardo is covered in skin impressions. And so we can get an idea of like how the scales literally looked and were shaped. So it's really fascinating to be able to get like ideas of what the actual body looks like versus just the bones. And then another really famous specimen that I think it was found around the same time, but was only more recently prepped and shared with the public, is another dinosaur called the Dakota fossil. And this is an Edmontosaurus, so another duck-billed dinosaur. That this one is like, they've got a good chunk of the dinosaur, but the most significant part that they have is one of the limbs, like one of the feet. And I mean, it is crazy looking. I think I've made you look up pictures before.
Yes.
And we'll be posting one. So this specimen is housed at the North Dakota Geological Survey, a great group. And it's crazy because not only is like the skin preserved in this limb, but literally like a hoof-like nail at the end, the distal most part where the toes would be. I mean, it looks like a horse, which a lot of folks have pointed out that hadrosaurs probably did have, this kind of herd, kind of ungulate-like behavior for dinosaurs. And because of mummified dinosaur fossils, we're getting a really good idea about what their limbs actually looked like, because it can be hard to reconstruct that from just the bones alone. We don't know, but because of these mummified dinosaurs, we do have a better idea of what the entire organism actually looked like. And like I said before, this is not the actual skin or hoof of the Dakota dino mummy. This is mineralized impression, right? Replacement. So if you were to sample it, you're not going to get like organic material out of it necessarily in this case. We'll talk about some weird examples. But is that like the case for this placenta that you're talking about too? Like we can see it, but what actually is it?
Yeah, so when people first excavated these beautiful yet sad mares with their little foals inside of them, and thought that it was organic tissue, the black stain that you see in the stomachs of these fossilized horses and the hair that you see on the primates and the skin impressions and the feather even, it's not actually original organic tissue anymore. It is exactly preserved as if it was, because what has happened is that you have these animals dying in this lake. They sink to the bottom where there's no oxygen. And the only thing that can eat them down there and decompose them is a type of bacteria that hates oxygen. They're called anoxic bacteria. And they produce a mineral called siderite. So if you zoom in and you look at the edge of these placentas, you look at the edge of the fur, you look at any stuff that is black and looks organic at Messel, it's not actually flesh that you're preserving. What you have is these bacteria, as they were consuming the placenta, they ended up precipitating the mineral siderite. And not only did they kind of create basically like this outline of these delicious organs that they apparently saved until last to eat, they also entombed themselves. So if you look at the surface of these placenta with a electron scanning microscope, what you actually see is bacteria that have encased themselves in siderite. Not technically organic tissue, but a very, very beautiful and very strange way of getting an imprint of that soft tissue. Weird, right?
Wow.
Yeah.
That's really weird. Like, again, like not a classic example of what is fossilization? You're like, well, sometimes bacteria go.
I mean, I do know that these kind of like bacteria biofilms, whether it's the actual bacteria or something that they're precipitating around things, they are often responsible for these beautiful cases of preservation because they basically like provide a shrink wrap that's like the distinct halo of these organs, right? Bacteria are surprisingly helpful sometimes.
It's a balance, even for us living creatures.
Yeah. But once you're dead,
us trying to get fossilized.
Well, I mean, really, as you're dead, too. You want the ones that will preserve you because they accidentally encase themselves in siderite or whatever. You don't want the ones that will just eat you. All right. So in the case of Messel, beautiful as it all is, it's not really organic tissue, right? So like no DNA. If there are proteins, they're probably the bacteria's proteins, not the actual horses. That's not always the case, though, is it?
That's right. In some cases and previous years, folks really thought that the longest that true organic material, like cellular material, could survive was maybe about 1 million years. However, there's this really, really, really famous T-Rex specimen that I had to spend a lot of time with in my past. That's a really, really cool specimen that tells a fascinating story about soft tissue preservation because the specimen is like 67, 68 million years old. We are talking the last of the dinosaurs, you know. This T-Rex was preserved in such a way. It's a long story, but when they excavated it, they had to use a helicopter to get it out of the field. And the helicopter could only carry so much weight. So they had to cut one of the jackets in half, and they had to cut one of the jackets that had the femur, which is the thigh bone in it, like right in half, which is, people are like, oh, the tragedy. And the reality is, guys, we break fossils all the time and then glue them back together. It's just how it goes. And this situation also provided an awesome opportunity for this researcher, Mary Schweitzer, to sample the inner parts of the bone before any of it had gone to the lab and been smothered in glues and all of these other like adhesives and whatnot. And she was actually able to identify straight up actual soft tissue in this thigh bone from this T-Rex specimen. So the T-Rex is known as B-Rex, and they were able to determine that it was kind of like protein-ish. Mm-hmm. Like she talks about looking at it under the microscope and being able to like poke the cell and having it still have some wiggle to it.
Wiggle to it?
Yeah.
Like gelatin.
Yeah, she literally, it was still soft and it was still transparent and still flexible. Yeah. And then they were able to actually identify this tissue even further, which is fascinating, as medullary bone tissue. And again, this medullary bone tissue is really significant too, that they are able to determine that because it's found within the bones of birds and dinosaurs. And it's only found in female birds that are preparing to lay eggs or have just laid eggs. So that means that B-Rex is not only the first dinosaur to be found with soft tissue preserved, actual soft tissue, it's also the first T-Rex to be conclusively shown to belong to one sex or the other. A lot of times with T-Rex sexing, they look at...
what a fun field to have chosen for yourself.
Well, they talk about their lips like it's going to be the savior of climate change. So I'm not that surprised that they also make a big deal about the sex. But before this, like, and still to this day, most of the sexing comes down to like trying to kind of show sexual dimorphism and that females would have probably been larger. But this, when you have so few samples, it's really, really hard to say with any statistical significance. So B-Rex is a big deal to be able to say 100% confidence this was a female and that she had just laid eggs or had were about to. Now, this paper has been controversial. There's been folks who've said that no, this isn't actually like a protein or a tissue that you're looking at. It's a byproduct of bacteria. However, the more and more research that Dr. Schweitzer does, it becomes pretty apparent that it really does seem to be protein and tissue related. And it pushes it back from being 1 million years old of soft tissue being preserved in, say, a mammoth to now about 60 68 million years old in B-Rex, which of course, when we would give this tour and show off the B-Rex teeth and talk about B-Rex, the inevitable next question was, are we one step away from Jurassic Park? So, what are your thoughts on that?
We are many, many steps away from Jurassic Park, unfortunately. So when we talk about proteins being found in things, proteins are not exactly the same thing as DNA, which is what you would need in order to recreate these dinosaurs. So if you imagine that DNA is like the story of an animal's existence, right? Like it's all the little pieces that make them up. Well, that story, like books, is not actually super stable over time. It breaks into little pieces, and the longer that it has been, the fewer and fewer extended pieces you might find. So you could absolutely look at B-Rex in that soft tissue and find some chunks of DNA. But instead of having a whole book, it would be as if someone cut the book into individual letters. So it's going to be a little difficult to stitch those letters together, especially since DNA has, 4-6 letters? Not very many different ones. So as far as we can tell, DNA has a half-life. It's not a particularly stable thing. And that half-life means that probably anything that is older than 1.5 million years old isn't going to have long enough sequences of DNA for it to be in any way, shape, or form usable, which is, probably for the best. I've watched those Jurassic Park movies. They don't seem to really work out super well for anybody. Even the dinosaurs aren't exactly having a great time. Now, if you're listening to me say all of this and you're thinking, oh, I thought we had gotten DNA that was older than that. Well, we have had a lot of papers published on DNA that was older than that. Wasn't actually correct. We have like papers that are like, I found this 50 million year old weevil and we found DNA. Well, I want you to imagine, how do you figure out what a DNA sequence is? And how do you pull that DNA sequence out of an organism? Because as you are doing those actions, you are also very likely to introduce, say, for example, your own DNA.
Yes.
Or the DNA of other things that have been sampled in that lab. So when you are using PCR, which is a genetic technique, I think it's polymerase chain reaction, something like that.
Sounds right.
Right? Yeah, it sounds right. It's probably not. But it's basically trying to amplify the DNA that you're finding in these samples, which means that if you accidentally drop any of your own DNA, any bacterial DNA, anybody else's DNA into the mix, because those are longer chains, they're likely to get really well amplified because you, presumably if you are sampling DNA of a fossil, are still alive. So your DNA is significantly longer. So all of these other big fancy papers, especially the ones that came out in the 90s, all of them have been disproven because it turns out that somebody actually dropped weevil DNA in something and it was living modern weevil DNA. One of the sad things about DNA is that it actually is destroyed in the same sorts of circumstances that are better at preserving things like soft tissue. Water is really bad for DNA. Water will help it break down. So if you want to make DNA longer lasting, you need to dry it out. But even then, if it's more than like 1.5 million years old, you're not really going to be able to do so.
So far.
Yeah.
People are very obsessed with these topics, so I'm sure that people will put stupid amounts of money into it, even though we could be putting that into, conserving modern species that face extinction. But why would we do that when we could bring back a murder chicken? It's my own frustration. And I love that. I think that that's an important thing to also just, again, spotlight is the methods of how you identify things and how you sample things really matter. That's why I was talking about Dr. Schweitzer sampling it in the field before they went back to the lab, because the minute it is transported a long ways and it goes through the lab process and gets cleaned up, and we use a lot of chemicals when we clean fossils, that would mess it all up. So how do we get the right ingredients to make this preservation happen?
Right. So, regardless of what you're trying to preserve, whether it's proteins from B-Rex, DNA from mammoths, whatever it is that you are trying to preserve, there's a couple things that you really need to do in order to ensure that this super great preservation happens. First off, You have to die and be covered in a way that things can't eat you. Because if things eat you, goodbye. You're either not going to be fossilized at all, or you're definitely not going to be beautifully, wonderfully, amazingly fossilized. So make it inaccessible or make it unpalatable. So that's where a lot of these like mummified dinosaurs, mummified ice age things, that's where they really get protected, because if you make it into nasty mammoth jerky, jerkify something and make it unpalatable. The other thing that you need to do is that bacteria, well, it's harder for them to break down things like jerky. It's not impossible. So you really need to do some extra stuff to make sure that the bacteria don't break things down. Because even if they leave that nice rind of siderite, it is still removal of the organic tissue. So if you want organic tissue, you have to ensure bacteria can't break it down. So to do that, you want to remove moisture, because that makes it harder for bacteria. You want to make it anoxic, so low oxygen, because that limits the number of bacteria that can get to it. And ideally, you want to make it like antiseptic or antibacterial. So this is something that amber does really well. Amber has, right? Yeah, well, think about trees. They don't really want to have infections when they're leaking resin, right? So resin is full of things that are antibacterial, which is part of why amber preserves things so well. Weirdly enough, they also have these proteins inside of them, resin does, or amber does, that are basically, they're kind of like plastic molecules. They're these really long chains of molecules, and they kind of glom together as the amber preserves. So it's like plasticizing something, basically, because plastic also...
The plasticizing.
Yeah. So they reject bacteria, and they make sure that it's like a nice solid lump. And that also goes into the organism, and it zaps out all that moisture. So amber, even though it looks like you have a beautiful mosquito in there or whatever, it's actually removing the moisture from that insect. So it's drying and desiccating it at the same time.
Oh.
So it's bug jerky inside the amber nodule.
So would you even have blood still left in that mosquito to be sampled? The amber would have been like...
It would not be a liquid. So you would have dried blood inside of there.
Okay.
Yeah.
Okay. Well, I mean, could still be promising, guys.
Yeah.
Could still be.
I mean, there are preserved mosquitoes with blood meals. They're not actually in amber. There's a couple that have been preserved flat in sandstone. And when they're sampled, you can see the iron, basically, that tells you that for sure that is blood. These are also, of course, way too old. However, they think they're like 40, 50 million years again. So you cannot get DNA from them. But, you can say, they've been little bloodsuckers for millions of years. And I guess that's something.
Yeah, that they've been horrible creatures for forever. I mean, it makes it easier to hate them when you know it's always been there.
So amber is a form of embalming, by the way, because it is desiccating things. Yeah.
Ooh.
Yeah.
Oh, okay. Well, good for you, trees. Good for you.
Yeah. So embalming, it's the process of removing moisture. Embalming, that's a big part of it. That's what you're doing with like that formaldehyde. You're trying to make it antibacterial and remove moisture so that the body sticks around for longer. But even with embalming, if you don't cover it up, the bacteria will eventually get to it, right? So you can't just leave these mummies or whatever they are on the surface and have them be protected for forever. If you really want organic tissue to stick around, you've got to make sure that things can't get to it. And that's a big part of it. Yeah.
Okay, so I mean, where exactly does that happen then? Okay, I guess we'll take a break, and then when we come back, we should go ahead and dive into where do the stars align for these processes to even occur. So we'll be right back.
If you're enjoying this episode, consider supporting us on Patreon. You can enjoy tons of bonus content for less than you'd pay for a bag of plastic quote-unquote dinosaurs that somehow always has Dimetrodon in it. You can also get access to outtakes, clips we couldn't quite fit, bonus episodes, and of course, you can suggest episodes for us to film next. Maybe one on how Dimetrodon isn't a dinosaur, for example.
Looking for other awesome paleontology podcasts? Why not check out Paleo Nerds featuring friends of the show, Ray Troll and Dave Strassman. And hey, both of us have guest starred on that podcast, so you can technically listen to us there too. Aren't you lucky? Welcome back. All right, back to dead things. So, How do we get this perfect, list of ingredients going for this type of really, really high level of preservation? So where does this happen, Megan?
Yeah. So if you really want to have great preservation, either of organic material or of, soft tissue in kind of general sense. I mean, we've already obviously talked about amber. Amber is a great one, but amber really only works if the animal is small enough that it cannot escape the amber and can be coated in it. So
right. I was wondering about that. You'd have to have a very large tree that was very productive of sap.
The ooziest tree.
Yes, the ooziest. Well, I am now just like, it's making me have even more respect for trees than I've already had to know that they're also like nature's coroners, thank you, like, wow, thanks for the oxygen and the preserving of dead things.
Yeah, so amber does preserve things like insects. It preserves things that basically can't get out of it because it's sticky. So no T-Rex is going to be found in amber. But there's also deserts. Deserts love to dry things out. Drink your water, folks. So they will very quickly dehydrate things and they can preserve things for long periods of time. Anoxic lakes and anoxic ocean environments, they can preserve some organics, but they can also lead to bacteria preservation, which isn't always bad either. And then bogs. Bogs and peat. Yes. Yes. They are very acidic, I think. They might be basic.
It's one of them.
They're one direction on the pH scale that is bad. So they're not a 7. They're not a 7. And because they're bad in whichever direction it is that they are, they can prevent bacteria from breaking things down, and they can preserve things really well.
It says that bogs are naturally acidic. You were right.
Oh, yes. Why did I undermine myself?
Yeah, exactly. Stick with your gut.
Bogs are another great one. But of course, if you really want to get organic tissue, look no further than your freezer. Or, nature's freezer, aka ice/permafrost, has a lot of stuff, doesn't it, Amy?
It does for now until it all melts.
Yeah.
So I should see, and then I laugh, and other people are like, that's not funny. And I'm like, but it kind of is.
Yeah, it's bad, but it is funny to be defrosting nature's freezer.
And now when I say ice, like, I think there's like that picture of like, the Ice Age little squirrel, like trapped in like the actual ice cube situation. That's not how permafrost actually works. Like permafrost is sediment, right? That has kind of the connective material is ice that's holding it all together. However, it is not just like a river of ice beneath your feet. That's not exactly how permafrost works. It is a phenomenon that has to occur in places that are far enough north to actually support it, to stay cold long enough. So this is where soil and sediment are frozen for at least 2 consecutive years. That's the official definition of permafrost. A lot of permafrost has been frozen for way longer than that. We're talking hundreds of thousands of years. And the upper part of permafrost is going to thaw in the summertime. And this is how we can still support like high Arctic forests and things like the tundra and whatnot. But the lower layers are going to remain frozen year round, keeping our little buddies nice and preserved in their icy, soily graves. When I was reading about permafrost, I had no concept that it can be 1,000 meters thick in some areas.
Jesus, that is really thick.
I know. And I was like, how do you know that? I'm sure it's like a geophysical kind of thing to be able to check out the specific type of density that you get.
Geophysics is just giving the Earth ultrasounds. That's what geophysics is. That's their job.
This is why my husband gets so excited during our ultrasounds. He's like, oh, these are seismic waves. And I'm like, yes. And then I'm geeking out about ontogeny. So then the ultrasound tech is just like, who the hell are you people? But yeah, geophysics is fun if you're into that kind of thing. Thank you for being interested, because I'm not. Yeah, so most of the places that you're going to find permafrost are going to be Alaska, also in northern Canada and the Arctic, and then Siberia, so the northern part of Russia as well. There have been some really, really, really famous permafrost mummies, if you will, discovered in the permafrost. Some of the most famous are all like different juvenile mammoths, most of them. We've got Yucca is a woolly mammoth, and then we've got Dima and Yuba. These are juvenile mammoths. They're all discovered in Siberia. Yucca is considered the best preserved woolly mammoth specimen in the entire world. She was approximately like 6 to 8 years old when she died.
So
and she was discovered by tusk hunters in Siberia in like the 2010s. So because there are woolly mammoths in the permafrost with tusks, which are ivory, which are still to this day extremely valuable, people go and they will hunt for the ivory of these fossilized mammoths. At least they're already dead.
Yeah.
And then one of my favorites. So when I was reading about this, we're going to talk about a specific specimen from Alaska that's famous for a couple different reasons. So to do this research, I ended up buying the guy's book on this. It's called The Frozen Fauna of the Mammoth Steppe, The Story of Blue Babe. It's by the paleontologist Dale Guthrie, who I'll be talking a lot about. And he goes into like the history of different permafrost animals. And by far and away, my favorite one was about this pony that was discovered in Siberia in 1968. And it was probably about 35,000 years old. It was found deep underground by miners, and they found it from the bottom up. They were like excavating their mine tunnels, and they found these dangling legs, and they were like, let's hang our cables from this naturally provided anchor into the ceiling. And so they have all of these like lights are all dangling from this Ice Age horses' hooves that are just dangling into the thin air of this mine tunnel. And then they eventually were like, okay, these legs are getting in the way. Let's excavate it. And some scientists came in and they were able to, they blasted the tunnel. And so that they had to like go gather horse limbs from the explosion site. But anyway, they concluded that like they never found the head. And they concluded that the head was probably never preserved, and that the head was probably ripped off, and then the rest of it was frozen. And Dale includes this great figure of this in the book. So I'm going to just hold it up. I have a picture of it too. Can you see this?
Oh, that's so stupid looking. You know, this is a podcast, so it's an audio format. So why don't you hold that up again, and I'm going to try and describe this graphic a little bit better.
This is good. And we'll post a picture of it too.
And I'm also only describing what I can see. So this is a 6-paneled cartoon, 3 on top, 3 on bottom, and there is a pony that looks kind of like one of those. I forget, like it's a Perwash for horses. It starts with a P. Perwalski.
Oh, yeah.
Yeah, whatever. It's a pony.
Yes,
and it looks,
those ones.
And it looks like one of the wild ponies. It's got like a very stiff-looking mane. So in panel 1, it appears to be walking on its hind legs in water. I don't think it is, but that's kind of what it looks like. It looks like it's maybe doing some sort of ballet routine. In panel 2, it has tripped. It has broken its leg, and now all 4 legs are underneath the water that I'm assuming is going to become ice at some point. Panel 3 is really difficult to interpret. It looks like maybe half of its head has been ripped off. And it does stay in the same body posture for the rest of the panels. Panel 4 and 5 and 6 all appear to have no head. Panel 6, I'm not really sure what's going on there, except that it looks like maybe one of those Mario flowers is reaching up to like ingest it from below.
Oh, and what you can't see in the last one is it has a little lantern hanging from its hoof.
And then they dangled things from its legs.
And it's emanating light into a tunnel.
I wonder why its foot got longer.
And yeah, it shows some sort of predator ripping its head off. Yeah, I'll read you the caption so that we can compare to what you came up with to what they wrote. They talk about how it is relatively well preserved and missing only the head and neck. The scenario of the death and the preservation that best fits the data is that the pony was mired up to its neck. Oh. Then the neck and head were eaten by predators or scavengers. Later, the body was further buried by the flow of silt and gravel down slope. The body was found from below in the ceiling of a gold mine drift, and its hind leg was used to attach cables and hang lanterns until someone realized it was the mummified leg of a Pleistocene horse. Period. Science.
Yeah.
Science. So that's definitely my favorite. So as you're gathering, these fossils are frequently found through people looking for them exclusively through like tusk hunting and then mining. largely gold mining. And that's actually how the specimen that I'm now going to talk about, which is the one from Alaska, which is called Blue Babe, was discovered. And the whole reason we bring this up is because, it's a question of how old will you go? And I want you to start thinking about what would be your limit of how old a piece of meat would have to be before you would say, no, I'm not going to eat that anymore. Because for this group of paleontologists in Alaska, their answer was, 50,000 years sounds fine. And they ended up actually eating part of this Ice Age bison that was discovered. And it's a fascinating story that I'm going to give you the highlight version of at this point.
How bad was their food poisoning?
Well, and that's the thing I want to talk about. I'll read you some of these quotes from the dinner party from hell. And I just, their arguments are sound. I just don't think I would go there personally, but I have IBS, so I don't need to do that to myself. I don't need to do that. But the story goes back to the late 1970s. A family mining operation was looking for gold in a placer mine. So what a lot of times they do is they use these high-powered pressure hoses to actually just blast away silt, gravel, clays that are around a creek or some sort of tributary of some sort, and to expose the gold-rich veins. And then they'll go through all of those sediments and they'll actually look for gold. And so that's how they blast it apart, and they find this mummy in there of this bison. They recognize that it was a bison, so they call the University of Alaska Fairbanks paleontologist Dale Guthrie, who I mentioned earlier with the book. He comes out, he's like, oh snap, this is a big deal. We got to get this bison out. It's summertime. This thing is it lasted 50,000 years and now it's going to rot in the modern day. But you got to think about how often that's probably happening too, when things just erode out naturally. And so what's really interesting is that the bison had a specific blue color to it. It's called from the mineral vivinite, and it just happens to be a byproduct of the preservation. And it has this really, really distinct blue color. So they called it Blue Babe. Again, babe being a wink, wink, Paul Bunyan kind of reference. Yeah, so it's vivinite. And I'll show you some pictures of it because they had the skin of Blue Babe was blue dusted, and then also around the horns was covered in a blue wart-like growth, which I'm like, yeah, let's eat that. Yeah, that sounds great. So it's the majority of this bison that's been preserved. I mean, it's a lot of the upper body, I would say, from shoulders through the head, a lot of some of the forelimbs, not a whole lot of, say, the torso, but a lot of the neck meat was still there, which is what becomes significant. Okay. So it was determined through extensive study. I highly recommend reading the book. It does not take very long to read, and it is full of little gems like that weird pony hanging from the ceiling. So he was able to look at the pelt, the lack of decomposition, the lack of bugs being preserved to determine that it was probably killed in late winter or early spring. Okay. He concluded that it was killed by a pack of Ice Age lions, the American lion, because the tooth and claw marks on the carcass match the dimension of the American lion. They also compared it to wolves and foxes and bears, but the actual dimensions of the bite marks matched to the best with the American lion. So they concluded that it was essentially, prehistoric feline fancy feast up in here. And so one of the questions is how did Blue Babe avoid decomposition, which is a question that we've been talking about a lot with today's episode, because you were killed by lions, you were eaten while they got their fill. So why wasn't the rest of you eaten? Why wasn't the rest of you decomposed? Mm-hmm. And he concludes that because the lions, eviscerated this bison, that actually allowed for the bulk of the mass of the bison to be spread out, which then led to the rest of it freezing much faster because it no longer had the thermal inertia of this really nice ungulate gut system keeping it super warm. And that's what likely kept the bugs from coming and eating it because it froze so fast after they gutted it.
Interesting.
He went to some weird experiments to determine this. Like really bad neighbor. He was a bad neighbor, but there was a herd of 15 bison that died of natural causes up, about 2 hours away in the Delta Junction area of Alaska. And so he calls up, Fish and Game or whatnot and is like, hey, can I get one of those bison for a personal experiment? And so he goes and he grabs one of these dead bison. They died from eating drinking fertilizer water,
weird.
Which is really sad.
Weird.
But he went and he got one of the bodies. He took it back to his house. He strapped it to a tree and then watched it decompose all winter. And he didn't do it. And that's how he concluded that something had to have gutted the bison, because otherwise it just doesn't make sense because his bison just got disgusting and totally decomposed and full of flies and all the fun stuff that comes along with bugs breaking down organic material. So that's how he came to the conclusion that it had probably been fully gutted, which is really, really fascinating.
Yeah.
But so then the absurd part of this becomes to the fact that they decided to freaking eat Blue Babe, which I'm horrified on a couple of different levels, which we can talk about all of them.
Yeah, let's do it.
So essentially this man excavates this bison. He studies it. He collaborates with scientists across the freaking world. He goes to Europe. He goes to Russia. He studies comparative specimens. He gets funding to make sure that Blue Babe is preserved. He gets this bison taxidermied, and it's still currently on display at the Museum of the North at the University of Alaska Fairbanks. It's a great museum. I've seen Blue Babe. It's a very impressive specimen. And after all of that work, he's like, now let's eat it. Which I just don't understand at all. And so he says, in his book, he wrote, all of us working on this thing had heard the tales of the Russian excavating things like bison and mammoth in the far north that were frozen enough to eat. So we decided, you know what we could do? We could make a meal using this bison. And they did. That was their whole plan. So let me describe this meal to you.
Yeah, what did they choose to make?
It was a very special occasion. Dale waited until his colleague, Eric Granvist, completed his taxidermy work on Blue Babe. Okay, so again, you just spent weeks handling this specimen and preserving it, and then you're like, yeah, I'm hungry.
I've been touching this thing for weeks. Let's put it in my mouth. Ugh.
And then they had another friend in town, Bjorn Curtin, who was there visiting, giving a lecture. And Dale Guthrie was like, well, let's have a dinner party. And he said that the abdomen area was too decomposed and there wasn't really a good source of meat except for the neck. So they decided to make a neck steak, which didn't sound very good, but we decided to add a lot of vegetables and spices to it so it wouldn't be so bad. And then he wrote, serving a table of 8. And I'm like, who were the other 5 people? You do not list them. I would like to connect with them and ask if they survived this meal. He took...
It's like a murder confession.
I know. It's such a weird dinner party, too. I mean, I know we said that we're weird guests at dinner parties, but this is taking it to the next level. He said he let the neck meat defrost. He said that as it thawed, it gave off the unmistakable beef aroma, not unpleasantly mixed with a faint smell of the earth in which it was found, with a touch of mushroom. My favorite. Oh, yeah. Oh, man. He said they added a generous amount of garlic and onions with potatoes and carrots and a little prayer to not get, dysentery and die. They also needed to drink a lot of wine, which I also think makes sense.
Yep.
And he said that they all said it was good and had an earthy taste to it. And when he was asked about well, were you worried you were going to get sick? He literally said, well, it would take a very special kind of microorganism to make me sick because I eat frozen meat all the time of all the animals I or my neighbors kill. And so he's saying that because he's this Alaskan outdoorsman, eating, wild game and caribou, that has prepared him for eating, the thing, like 50,000 years old. So apparently this dinner party was a big success. No one got sick. I don't know if anyone had seconds. These are the questions I would love to ask. But they decided to eat their specimen. And so that's where I get conflicted, is like, A, I think it's a health concern. I don't think that that's a good idea. And then B, it's like you have one specimen and you decide to do something that cannot be repeated ever again. Like that's the collection manager in me is going, why? But now no one else can study that neck meat, So those are, I think it's a fascinating story. I would not have made those same choices. And I do just have some questions about why.
I feel like
happened.
I almost get it. Like, oh, it's cool that you could eat something that nobody else theoretically could. But also, I learned at a young age to not put everything in my mouth. Bugs, they're the stomach acid of the pond world.
They're like trees, right? Of bogs, of plants, of swamps, of, yes.